Hello everyone. Today we will be looking at genetics and how they pass from parents to offspring. So this is chapter 9, Patterns of Inheritance, Mendelian Genetics. The first person we talk about is Gregor Mendel. He was a monk in what's now the Czech Republic in the mid-1800s. So really we haven't known about genetics and the patterns of inheritance for very long. Mendel became a monk in order to do science research. Back then, the best way to go about doing different scientific research was to become a monk. He looked at plants, mostly pea plants, and he looked at how traits were passed down. He'd take different types of peas, different colored leaves. He'd see what traits their offspring would have based on different colors, and then he'd breed those together, see what traits they had, and so on. People before him obviously noticed that offspring were like their parents, but he didn't know how to predict traits being passed on. They didn't know, and Mendel figured it out. So here's a pea plant that he looked at. They have lots of different traits, and they are super easily manipulated, and they can self-fertilize or they can be fertilized. So what Mendel, here we go. So what Mendel did, he cross-fertilized, which means he took pollen from one plant, put it in another, so that those offspring would be half the white flower and half of the purple. So here's the white, purple, and this generation is called the F1 generation. The first group is the parent group, and their offspring we call the F1 generation. So parents are up here, offspring down here. He also created true breeding varieties, which means they only had one trait. So if they were purple flowers, both versions of their chromosome were purple flowers. He bred those together, and he could see that offspring were all purple. Therefore, he knew that they had to be true purple. And then he would do the same thing with white to make true white. He'd breed the white flowers together so that all the offspring were white, and he'd know that's true white. And then he did a cross where he'd cross the true purple with the true white and look at the offspring. How many were purple and how many were white? And then he'd breed those offspring together. So you see why we can't do this on humans. You don't really want to be breeding offspring together. Really don't want that. But in plants, it's all good. When he bred the offspring together, he'd get the F2 generation to see how many were purple and white then. So we have the parent generation, we have the F1 generation, and then when we breed the F1s together, we get the F2 generation. Mendel looked at other pea plant characteristics as well, like seed shape, seed color, flower color, pod shape, and pod color. These provided a lot of traits that he could test to see what would happen. So he also looked at some more characteristics, like if they were terminal, so if the flowers were on the end, or axial, if they broke off the main axis. And he looked at if they were tall, so stem height tall versus dwarf. So lots of different traits here. Oh my goodness, I keep clicking through this. Okay, so now we move on to monohybrid crosses. So mono means one. First, Mendel would do a monohybrid cross. So this is what we were talking about before, where you breed the true purple with the true white. Mono means one. When we do a monohybrid cross, the first generation are all purple flowers. So that told Mendel that the purple gene masked the white genes. The purple overshadowed the white. Then he wanted to see what would happen if he crossed them again. So all of these flowers in the F1 generation right here, so F1 generation, all these flowers, have one gene for purple and one gene for white. And what he found with the F2 generation was that 3/4 or 75% had purple, and 1/4 or 25% had white flowers. And by finding these ratios, he was able to predict what was happening. The 4 hypotheses that he formed all work hand in hand, so we don't have to say that just one is true. He hypothesized that there are alternate forms of genes called alleles. Different forms, they came in different forms, like an allele, like there's an allele for purple, there's an allele for white, and for each characteristic, each organism has 2 alleles. We talked about how we have 2 copies of each chromosome, one from mom, one from dad, and all those have the same genes but can have different versions. Well, that's what Mendel's thinking here. So to go back to the flower example, on each of the chromosomes, they've got two alleles, so one for purple, one for white, or they'll have 2 alleles that are both purple or 2 alleles that are both white, just depending. Then gametes, so sex cells, only have one of those alleles, so that when they combine together, so an egg has one allele, a sperm has one allele, so that when they combine together, you've got 2. And these alleles, or again, variations of genes or gene versions, can be dominant or recessive, so they can mask the other. So if they mask the other, it's dominant. And if they're able to be masked, if they're able to be overshadowed by the other, we call those recessive. And I'll talk more about the difference between dominant and recessive here in a minute. So how does this happen? Mendel made a Punnett square to predict offspring. This little square right here, called a Punnett square. On the top, you have one parent's genetics. So up here, and then on the bottom right here, you have the other parents. Each of these Y's is an allele. So each of the Y's you see is an allele, a version of a gene. What we do for each trait is we give it a letter. So here it looks like we're doing pod color and we chose the letter Y. If the letter is capital, that means it's dominant. And then lowercase means it's recessive. So if we have a big Y and a little y, you'll see the dominant trait prevail, which is the big Y. You can see the big Y here stands for yellow, and the little y stands for green. So we chose Y because yellow is dominant, as you can tell by its capital letter, and the little y will stand for green. If you see big Y, little y right here, the pod is going to be yellow. You'll only see green in the little y if you have 2 copies of it. That's what makes it recessive. So right here you can see the pod is green, little y, little y, little y, little y. This is recessive right here. So the green comes out. So what you do when making a Punnett square is for each allele, there's a 50-50 chance of sending them. So on the top, you put the big Y, and on the bottom, the little y. Then you go from the top down. So big Y here, little y here, and then top down, little y here, little y here, and then you drag that across too. So you're just big Y, little y, little y, little y, big Y, little y, little y, little y. Inside of the Punnett square, you now see 4 possible results of the offspring. You look at the top 2, big Y right here, or big Y, little y, little y, and they look yellow. The big Y is yellow, the little y is green, yellow masks the green, and the plants look yellow. These that have 2 little y's look green, so 2 out of 4 here are yellow, 2 out of 4 are green. So 1, 2, 3, 4, 1, 2 are yellow, 1, 2 are green. So we'd say there's a 50-50 chance they'd be yellow or green. So 2 out of 4, 50-50. Here's another example with that same flower color from earlier, purple versus white. Our purple flower is big P on the left right here. And the white flower is little p. So big P for purple, little p for white. What happens is the purple flower sends a purple allele, which is dominant, and the white flower sends a white allele, which is recessive. As you can see right here. So all of their offspring have a purple dominant allele. So that's the F1 plants. So they look purple. This is their phenotype. Phenotypes are observable characteristics that we see. So purple would be the phenotype. In their genetics, they have a purple big P and a white little p copy. And this is called their genotype. So big P, little p would be the genotype. The color purple would be the phenotype. Next step is to cross the F1 plants. So the big P little p with big P little p. Take those together, we make a Punnett square. And what you see here is that 1/4 is little p, so right here, or white flower. And what we'd expect to see is 3 purple flowers for every 1 white flower. To see this pattern, you'd have to breed a lot of plants, and we wouldn't really see it just breeding a handful of plants. In science, remember, we need to do a lot of trials to ensure good results. And what Mendel found was a 3 purple to 1 white flower ratio. On our last chance, we had a 50-50 chance. This would be called a 1-to-1 ratio, equal amounts of yellow to green. Whereas here, we have a 3-to-1 ratio. For phenotypic, 3 purple to 1 white. When we look at genotype, we see 1 big P, 2 big P little p's, and 1 little p. So we'd have a genotypic ratio of 1 to 2 to 1. So 1 to 2 to 1. So now let's look at what I mentioned in the previous slide. So the differences between genotype and phenotype. Phenotype is an organism's physical characteristics, so what they look like, hair color, eye color, ability to roll your tongue. These are things that we can see. Genotype is the genetics behind it, so what alleles they have. For white flowers, the phenotype is white, and the genotype is little p. You can tell an organism's phenotype by looking at it, but you can't necessarily tell its genotype, because remember, there's the one that has big P, little p, that would still look purple. So if the flower is purple, you wouldn't know if it's big P, or big P, little p. There's no way to know that. So moving on to Mendel's principles of segregation. This states that those alleles separate when we make sex cells. So that the sex cells, the sperm and the egg, only have 1 copy of each. Here you can see we start at the middle step at F1. Remember, we have parents F1 and F2, so F1 would be the middle step here. And this shows how there's a 50-50 chance of sending out each allele. Only one gets sent out from each parent, and they can go to either. And then moving on. Alright, now we're going to get a little bit more complicated, but it's not that bad, I promise. A dihybrid cross is different from monohybrid. Di means 2, so here we cross 2 traits. They have 2 characteristics that the parents are true breeding for. So the first generation parent, for example, in this one, little r, big Y, the seeds are wrinkled and yellow. And they have 2 alleles for wrinkled and 2 alleles for yellow. So little r, big Y. The others are round. They've got 2 alleles for that, big R, and they are green. So little y. So the capital R, we're talking, looks like it means round. The little r, wrinkled. The big Y, yellow. The little y, green. These are going to send out an R and a Y each. So a little r and a big Y, and then a big R and a little y. In the F1 generation, all of the offspring are going to look the same. They're going to look round and yellow. Notice big R, round, big Y, little y, yellow, big R masks the little r, big Y masks the little y, so that's why we see this as round instead of wrinkled, and this is yellow instead of green. And that's what we see in the F1 generation. But then we'll cross the F1 and we'll get complicated. So take the F1, multiply it by F1. So on this side you see big R, big Y, so big R, big Y, then big R, little y, big R, little y, little r, little y, little r, little y, and little r, big Y, little r, big Y. So we're just using every combination we can make, and then same up here. Notice that these are the same right there. So these offspring, or I guess they're the parents now, can send out one trait, round yellow, round green, and wrinkled yellow, to just put the genotype into phenotype wording. And there's a 1 in 4 chance for each of these. So it's random, and each offspring will look different, is what that's saying. And then same with the top again. Notice that these are all the same. So what we have then is different possible traits out of these 16. So then we just cross all of these together, so connect the dots kind of. So R, R right there, Y with Y right there. Same down here, make it look a little bit different. We take this with this, so put the R's together, put the Y's together. We get this wrinkled in yellow. But anyways, out of these 16, so 4, 8, 12, 16 squares, 9 have round yellow offspring. So 1, 2, 3, 4, 5, 6, 7, 8, 9 are round and yellow. And this is the 9 in this 9 to 3 to 3 to 1 ratio. So 9 of them are round and yellow. The 3 here depicts the 3 that are round and green. So 1, 2, 3 we see are round and green. The next 3 is for the one, where is it? Here we go. The next 3 is for the 3 wrinkled in yellow. So wrinkled yellow, wrinkled yellow, wrinkled yellow. That's 3 right there. And then the 1 right here is for the sole wrinkled in green, P. And that is what we call a 9 to 3 to 3 to 1 ratio, and this will come up a lot throughout genetics. And if you had 1,000 offspring and looked at the different traits, you'd still find this ratio when we do a cross like this. So now let's throw it back to homologous chromosomes. Remember, this is when we have 2 copies of the chromosome, one from mom, one from dad. They have the same genes, but they can have different alleles or different versions. Those alleles are on specific locations on the gene, and we call these specific locations loci. Locus is singular, loci is plural, so if you see L-O-C-U-S, it's just talking about one singular loci. Loci is plural. It's talking about a lot of different locuses, but we don't say locuses, we say loci. But anyways, moving on from that, here you can see the different versions on the same locus with allele A right here. A, let's just say that this is for eye color. The big A could be for blue eyes, and the little a could be for green eyes. It's the trait for eye color at the same location, but there are different variations. Here's an example of different genes at different loci with different possibilities. So the big P right here, has two copies of the same allele. They're both dominant. And then little a has two that are both recessive. So right there. And then big B, little b has one dominant and one recessive, which is what we call heterozygous. The other two we call homozygous, and homo means same. B has the two copies that are the same. So big B, they're both the same. We call these homozygous dominant. Little a, homozygous recessive. And then big B, little b, we call heterozygous. Hetero is different, homo is same. So that's a good way to remember the difference here, just by looking at the letters. So with heterozygous, again, just to reiterate, the big B is, in this example, is the dominant allele. The little b is the recessive allele. So what we would see is the trait for the big B, because dominant overshadows the recessive. All right, a little bit more information on homozygous versus heterozygous. A cool thing that you can just read through these definitions, but a cool thing that sometimes happens is you get incomplete dominance, where when you have a dominant red plant like this, homozygous dominant, and a recessive, homozygous recessive white plant, mix them together, you get a pink flower. Again, this is a special circumstance called incomplete dominance, and I will talk more about that towards the end of this chapter. All right, we talked about the segregation of chromosomes, and this is another example of a dihybrid cross. Two traits, so coat color and vision, and different proportions of each is what's being looked at here. When we talk about random events in evolutionary theory, this can be part of it. You have random chance for passing on different traits you have to your offspring. So 50-50 chance for each chromosome getting sent on to your gamete or sex cell. Sorry. How you calculate probability is not complicated. You look at different events that must occur for something to happen. Let's say you want to know the odds of the recessive B. So little b, little b, little b, little b right here. What you do is you take the odds of each thing happening. So there's a 50-50 chance, a 1 in 2 chance that you'll have a little b. A little b here looks like it stands for tails. So the tails on this left coin, right here, there's a 50% chance of that, and then there's a 50% chance of getting a tail on this other coin. So what we do is we multiply that 1 in 2 chance, so 1/2 by 1/2, and we get 1/4. And we see out of the odds here that one of those 4 squares will be a little b, little b. If we're flipping 3 coins, it'd be 1/2 times 1/2 times 1/2, that they all be tails. So this will give it a 1 in 8 chance. So 1/2 times 1/2 times 1/2 equals 1/8, which means 1 in 8 chance. There are several disorders that are caused by just a single gene, so one protein in its code. So we'll look at some examples of those. Most disorders are recessive. Generally, if you have one mutated gene, that's okay, because as long as you have one that's functioning, you won't have that disorder. Here you can see that as long as one allele is dominant, you won't have deaf offspring. But notice the heterozygous individuals, they have one recessive allele, so depending on who they mate with and have offspring with, they could potentially have a deaf offspring because they're carriers. They're not deaf, but they do have that recessive allele that would make them carriers for deafness. The homozygous dominant does not have that recessive allele, so it is not a carrier. There are some dominant disorders as well, although these are very rare. For example, achondroplasia, this type of dwarfism, is dominant. So this individual here in this example needs only one copy of that trait to be a dwarf, and then he has a 50-50 chance of passing it on to his offspring. Some patterns, though, are not explained by Mendel's principles. I mentioned this a little bit with the pink flowers. Remember I called it incomplete dominance? We don't have the dominant masking the recessive. Instead, when we have heterozygous hybrids, they're pink. We can also have codominance where we see both, but we'll look at that with blood cells. So with incomplete dominance, you take the white flower, you take the red flower, you mix it, you get this pink flower. With codominance, if we mix the red and the white flower, the flower would be red with white spots, for example. So we would see both of those colors. They wouldn't be mixed together. Blood groups are an example of codominant. We have different possible proteins on the outside of our red blood cells. So that's what's determining our blood type. So we got 3 different versions of genes possible. We've got a gene IA that codes for A protein, AB, right here, IB, that codes for B protein, and a little I gene that's mutated. It doesn't code for a gene at all. Each person gets 2 alleles. They can get 2 copies of the same allele or 2 different copies. Think about combos. So AB with A and B. You'd have a type O blood. If you had a little I and a big I with an A, you'd have type A blood. But you could pass down either to your offspring. So to make this make a little bit more sense, on type A blood you have A proteins. So you either have 2 copies of A or 1 copy of A and the mutated little I copy, that would be O. Same principle with B. You have B proteins, so you either have 2 Bs or you have 1 B and 1 O. Type O blood, people with type O blood types have neither protein, and people with type AB blood types have both A and B proteins. And the picture below shows different expressions and different phenotypes based on the genotype. What's really cool with blood groups is that if you are, let's say you are a blood group and you have certain proteins on the outside of your blood cell, anything that's not on the outside, your body is making antibodies against. So if you're type O, you'll have anti-A and anti-B. So if you had a blood transfusion and someone gave you type A blood, type B blood, or type AB blood, what would happen is those antibodies right here, the anti-A and anti-B antibodies, would attach to the red blood cell proteins, and they clump them up and it cause death. So that's why it's super important to know your blood type if you're getting a blood transfusion or giving blood donations. Then type AB blood, they have both proteins on the outside, so they don't have any antibodies in plasma. Makes sense, right? They've got the antigens, the proteins on their red blood cells, and that makes type AB blood the universal receiver because it won't react with anything. Whereas type O blood is the universal donor because it doesn't have any proteins on the outside to react with. We've got multiple alleles, multiple options for genes. So this shows the genotypes we just talked about. So IA, or IAI for type A blood right here. Same with B. IAIB for AB, and then just little I for O. The antibodies we just talked about are in this column right here. And now this right here shows what would happen if we were to mix different blood with different antibodies. If you're type O and you mix with type A, type B, or type AB blood, the antibodies present would clump it. So notice how clumpy it looks here, and it only looks normal when it's got that type O blood. A, so notice how normal they look. So this A is mixing with O, it's fine. It's mixing with A, it's fine. Because you have anti-B, you can only not receive from blood that has B proteins, like type B and AB. So if we tried to mix A with B and AB, it would clump up, like in these two examples. Then we have AB, that is the universal receiver because it doesn't clot with anything, and it's not making any antibodies. So antibodies present in blood, AB doesn't have any, so it is able to accept blood from all these different blood groups without clotting. So see, it all looks normal, it all looks good. And as we discussed, this is codominance. Here we see the A proteins and B proteins in AB blood. And in flower, we'd see red and white spots instead of a mixed pink color. Both alleles are expressed in the phenotype right here. We've also talked about sickle cell anemia, where you have a single wrong amino acid in a primary protein structure, and you have this oddly shaped hemoglobin that causes this oddly shaped red blood cell that affects the ability to carry oxygen. It blocks blood flow to different areas, so that singular gene causes more than one problem. And when that happens, we call it pleiotropy. That's affecting one or more characteristics. One gene affecting one or more characteristics. Sickle cell anemia. Let's look more closely at sickle cell anemia. So an individual is homozygous for sickle cell allele. Hemoglobin's affected. Abnormal hemoglobin crystallizes, causing red blood cell to become sickle-shaped, which we just talked about. And these sickle cells can cause different issues that lead to different homeostatic imbalances. For example, different organ failures that can be fatal, and so on and so forth. You can just read through this area of the chart. Then we have polygenic inheritance, which on the other hand is multiple genes affecting one characteristic. For example, skin color. There's a very wide range of pigmentation levels, the amount of melanin your skin makes from very light to dark, and that's based on several genes together. And along with our genetics, the environment plays a big role on our phenotype. For example, height and weight can be determined by our nutritional content as well as our immune response and our genes. An example is rickets. We've all heard of rickets, where kids don't have the nutritional requirements of enough calcium and phosphate to put down their bone, make it hard. So instead, their bones are softer, which could cause them to bend, which would cause deformations to occur as they grow. So this is a little page to remind you there is theory of inheritance that states the following. genes are on chromosomes, and the behavior of chromosomes during meiosis and fertilization accounts for inheritance patterns. So this is everything we've talked about so far. We get interesting things happening when we look at our sex chromosomes, the X and the Y. Females are XX, males are XY. Males have sperm with a Y chromosome. So males are the ones that are the ones determining sex of the offspring. Think back to medieval times when kings beheaded wives for not bearing sons. Well, it was actually the king's fault, not the woman that he was beheading, because she can only give X's, whereas the male can give a Y. What's pretty cool is that in each cell, only one of the X's is active and the other is inactive, and it's called a bar body. What's interesting is that because males only have one X, they are much more likely to get X-linked diseases. So X-linked or sex-linked mean the same thing. They only have one chance at a good allele. So if they get a mutated version of an X, they'll get that disorder, whereas females get 2 chances. So let's look at sex chromosomes a little more and the traits that they can influence. This shows how the males determine the sex of the offspring. So here you can see we have 22 normal autosomes, and females have 22 plus the X, whereas males get 22 plus the Y, and either an X or they pass on either an X or a Y. This is the same in humans and fruit flies. Although in fruit flies, the XX is the male and the XY is the female, which is kind of weird, but we're not going to get into that at all. Just know that XX is female, XY is male. Let's look at some of those X-linked genes and the X-linked recessives I mentioned. So here you'll have a mother with normal X chromosome and one with the altered gene. So X chromosome with altered gene, X chromosome with usual gene. She is a carrier for whatever disease is happening here. The father has a normal Y chromosome and a normal X chromosome. So he's on this side, X and Y. If the mother has one bad X and she passes it on to her son, you'll have an affected son. See that right here? She can have a daughter that's a carrier, or so one normal, one affected, or she can have unaffected daughters and sons. So to put this into perspective, one example is color blindness. So one example of an X-linked or sex-linked gene. We have cone proteins in our eyes that take in color and tell us about it. So light-sensitive cells, those are those cones in our eyes. So here what you should be seeing is a 7. If not, you're colorblind. You can't see red versus green. Unless someone takes this test, they won't know if they're colorblind. They just see things a bit differently and may have problems distinguishing between different colors. With this X-linked recessive, if a male is colorblind, he had to get that from his mother. If a female is colorblind, her dad has to be colorblind because she has 2 bad copies. So she got 1 X from her dad, 1 X from her mom. So dad has to be colorblind. And the last example we're going to talk about is hemophilia. This is a disease where you can't properly form blood clots. So blood loss is severe This happened a lot in old royal times where they had a lot of breeding between relatives and cousins, so that they would keep all the wealth and land in the family. Pretty gross, right? So here you saw female carriers, and if they had sons, with only a bad X, they wouldn't be able to clot properly, and that would be fatal. So this is the end of chapter 9. I hope you enjoyed learning about genetics.